Let's talk about DNA adducts. What is a DNA adduct? I think most people probably will not be familiar with that term. Are these adducts actually attached to our DNA, to our mitochondria? What damaging or deleterious effects do they have on the body? And then do you see higher levels when you're doing this testing, the IGL testing? Do you see higher levels in sicker people as compared to healthier people? Or does everyone have very similar levels? Why do some people maybe have higher levels than others? We just talked about some of that. Is it potentially because some people also have less than optimal ability to detoxify, maybe overlapping in some way with methylation efficiency? Or is the primary driver of our level of DNA adducts really primarily related to our external toxicant exposure?
Some and all of that, if I can take it maybe from the last question back, it's a very complex interplay. And I'd hope that we can get to some of the intricacies later on. I think primarily in people that have a chronic, especially if it's a treatment-resistant condition, I do see much higher levels of adducts as a by far dominant observation, compared to sometimes we've got people with extraordinary symptom burdens and the adducts are not that bad. And then you need to go look elsewhere. Comparing as a general statement, yes, people that have clinical conditions where the body doesn't function and regulate well, the adducts are much higher, by 30, 40% higher, as compared to not that many, but I've been able to, or I've had requests from people that have a healthy life. They don't have a symptom burden. They think they're normal in terms of health status, where the adducts are usually in the normal range, which, by the way, gives me quite a bit of confidence in the range, the reference range established by the lab, that I do see typically, with few exceptions, that people that feel healthy and function and regulate well, they don't have much intracellular toxins, but they do, but in compared to patients that have challenges. And then, in terms of your question, what is a DNA adduct? It is the definition is a typically exogenous, but it can be endogenous generated chemical that is covalently bonded and reacted into the DNA double helix, specifically into gene regions, or the regions adjacent to gene regions that controls the gene region. I'm talking about the gene suppressor and gene promoter regions. It used to be, we thought it was just nonsense DNA, just kind of placeholders, but it seems like they're actually really important. And if you think of the effect of an adduct on the gene promoter region, you're going to make excessive amounts of copies of a DNA gene region when you're basically pushing the Xerox button to make copies of whatever's on the machine, right? So thinking about what DNA adducts are is it's a chemical that is bonded into a DNA gene region. A DNA gene region is something that has a message. It's a set of instructions of how to, right? So the gene region for MTHFR, most people know what that is and does. The gene region for the vitamin D receptor, or COMT, or for, you name it, the detox system, blue eyes, blonde hair, things like that, right? So when you have a chemical covalently bonded into the structure of the gene region, it causes several things. So if you can think of just immediately the message or the operating manual instructions for how do you make glutathione, or how do you make GSTM, the enzyme that puts glutathione to work, that instruction is distorted and essentially heavily redacted. And if you think of what that means in terms of downstream, and maybe we can talk about that in a little bit as well, what does it mean for cell health, cell function, that transcription through to translation into the metabolome, proteome, lipidome doesn't happen as per, kind of wild type code? But at the DNA level, it means the DNA, you've got typically a single strand break from the bulky chemical adducts. And not only does it distort the DNA message, but it most certainly distorts the duplication of DNA with mitosis and meiosis. And that's where the transgenerational problems come in, where you have junk DNA with or without the chemical transferred to other cells. And in our lifetime, we're thinking neurodegeneration going into cancer. And on the longer timeline, we're thinking of junk DNA, non-functional DNA gene regions being gifted to our offspring.